and lysine 9 of histone H3 trimethylation. These modifications are not only hallmarks for tracking the development of aging, but are also possible targets for anti-aging therapies. Non-coding RNA and chromatin remodeling are the other components of epigenetic mediators that are involved in gene transcription regulation. Literature shows that multiple nutrients and nutraceuticals play an important role in curing the aging process. The consumption of many nutrients that reduce oxidative stress, lowers chronic inflammation, supports genomic stability and DNA repair, including fish, fruit, and vegetables, suggest that a healthy diet and lifestyle can help to slow down epigenetic age acceleration. Maintaining good nutritional status will contribute significantly to improved health outcomes in terms of age-related disease and lifespan. This book chapter highlights the molecular mechanisms associated with senescence and aging, with a particular focus on the role of epigenetic regulation. Also, the effect of several dietary components in reprogramming age-related epigenetic changes and rejuvenation will be discussed. Aging and molecular mechanisms. Clarifying as well as reducing the processes of aging and the incidence of age-associated disorders have become a global problem. Aging is a complex process, carried out under the umbrella of multiple molecular mechanisms. ROS are considered the most acceptable mechanism for aging that involves the production of various radicals and peroxides. When these species exceed their antioxidant level, interact with the proteins, nucleic acids, and lipids within the cell, and this leads to oxidation that in turn results in cell permeability changes, DNA damage, and cell membrane destruction. In addition, a high ROS level also induces p38 MAPK activation that proceeds to senescence and finally aging. All the molecular mechanisms are interconnected, and it is hypothesized that an individual mechanism not only contributes to aging, but their connections play a vital part, as shown in Figure 1.1, which can be found in the print and ebook versions of this book. The signaling pathways associated with the molecular mechanism of aging are discussed briefly. Nuclear factor kappa B, NF-kappa B, signaling pathway. The activation of transcription factor NF-kappa B is involved in the transcription regulation of genes associated with cell proliferation, differentiation, inflammation, immunity, aging, and apoptosis. Senescence-associated inflammatory factors could stimulate the NF-kappa B signaling pathway activation. The NF-kappa B, upon activation, enters the nucleus and attaches to the genes triggering the immune response. In addition, the activation of NF-kappa B also regulates aging-related signaling pathways, owing to various senile degenerative disorders. That is, in Alzheimer's disease, an increased activity of NF-kappa B was found. Furthermore, the accumulation of beta-amyloid plaque is also associated with NF-kappa B activation and the release of nitric oxide, NO, resulting in increased oxidative stress. An autopsy study revealed that the NF-kappa B level in Parkinson's disease patients was 70% higher than in normal, healthy individuals. This suggests that the disease's pathological mechanism is related to the NF-kappa B activation. WNT beta-catenin signaling pathway. The WNT beta-catenin is a conserved signaling pathway with a diverse set of biological activities. This pathway has been considered to execute a significant role in regulating cell aging, and its activation in mesenchymal stem cells could lead to senescence phenotype. Literature has also shown that the activation of this pathway is related to the p53 protein overexpression and DNA damage that ultimately result in stem cell senescence. This molecular pathway is also associated with oxidative damage of DNA that further contributes toward the hematopoietic stem cells aging process. In the context of human physical aging, skin aging is the most important manifestation externally. A study in this regard showed that in the tissues of aged skin, The WNT beta-catenin signaling.
To keep listening to this audiobook, I've left you a link in the description so you can do it at no cost thanks to Audible's 30-day free trial.